everybody, my name is Mike Banerjee. Today I'm going to do my Passion Project 2021. Let's hear for me. I'm a pizza lover who enjoys reading science books. I also like Minecraft. I personally connect with the first paragraph of the IB mission statement because it can create a better and more peaceful world through understanding the unknown. My big idea is genetically modified foods. Why genetically modified food, you ask? Genetically modified fruits and vegetables point to benefits such as higher crop yields. Scientists genetically engineer fruits and vegetables to introduce resistance to diseases or pests. However, there are some disadvantages too. These are my conceptual questions. They help me build my lines of inquiry and they also help me in my inquiry in other ways. The TDT I chose was how the world works. Since with genetic modification, we are tinkering with how the world works. My central idea is finding out about genetically modified food may help people to take action for change. These are my possible learning outcomes. These are my central agreements. They help me organize my inquiry and they also help me create some rules. My logo is for an infographic. It is made for people to know more about genetic modification. The infographic color half orange and half deep blue focuses on modification in DNA. My slogan represents that it is the time for change. This is my organizing inquiry. The arrows have 2 points, which means, for example, while discovering more, you can still stimulate curiosity and vice versa. In the first stage inquiry, I tuned into the unit through KWL. In the second stage of inquiry, I used the Golden Circle graphic organizer to understand the why, how, and what of genetic modification. This is the structure of DNA. Inside the plant, there's cells. Inside the cell, there's nucleus. Inside the nucleus, there are chromosomes. Inside the chromosomes, there are genes. This is my understanding on a blog post. The striking feature is that I actually learned about E. coli bacteria being modified to make insulin. That was certainly new knowledge to me. This is a map of genetic modification. Red means no formal GMO-free platform widespread in use in most areas. Yellow means law restricting GMO in most areas. Green means GMO-free or national bans. I can see only Russia and some other countries and Europe and Africa and South America actually are GMO free or national bans. Either way, this is a TED Talk video reflection to peel the fruit. 1 is outside skin. Describe what you see in a TED Talk. GM foods can be hazards to both the environment and us. 6 is the heart of the story. What does the author want you to learn from the story? That GM food should not be ignored. You should, if you make GM crops, separate them from the regular crops and do studies with people who are not biased towards any particular company. This is my movie reflection, Jurassic Park. At some point in my learning journey, I took some time to pause and reflect. Initially, I was very excited about investigating GM foods. When I saw its actual impact, I was puzzled. Further research and mentor meetings and guest lectures helped me to think about my responsibility. Finally, I was happy to be done with the exhibition. I could match my inquiry process with lots of relevant information. During my research, I found out that all GM crops are banned. The only GM crop allowed is BT cotton. In my guest session, I also found out that BT bacteria is used to kill insects with an enzyme tailored to the insect digestive system. The insect is killed by the enzyme, but the insect is eaten by the birds, which disturbs the ecosystem. India is a highly populated country, so we need more food to sustain it. I did extensive research on GM foods. My research further helped me to understand how to identify GM foods. These are my assessments. I learned later on that GMOs are not as bad as they seemed from my research, so I made a post to show that GMOs are not as bad as seem. These are the tools and strategies I use, also known as NESW: Mind Map, Golden Circle Strategy, Timeline, Freya Model, Peeler Fruit, KWL, KOD, and Learning Pit. Here's a gallery of some pictures during the exhibition. These are citations. I like to acknowledge Ms. Chandrani Banerjee for being a mentor, Ms. Bushra Khan for being a PYPC, Mrs. St. Xavier for helping with ICT, Ms. Samikar for teaching us how to make a logo, Dr. Jayant Ravindra for being a guest speaker, and Mrs. Sakshi Lamba for being a guest speaker. Thank you.